السلام عليكم ورحمة الله وبركاته. This is the second lecture in your dosiform design course. First lecture was about the physicochemical characteristics of APIs or active pharmaceutical ingredients. Today we'll talk about physicochemical characteristics of the second component in any pharmaceutical formulation or any dosage form, which is the excipients. So in this lecture, we should uh, explain how to define the term excipients. These are the information that you should take from that lecture, should be able at the end of the lecture to define excipients. Explain the rationale of including excipients. Why do we have excipients in the pharmaceutical formulations? And then we should know the different functions that excipients will perform when they are added to the pharmaceutical formulations. And we should give examples of excipients belonging to different functional classes. We should recognize the content of excipient monographs in compendia and in other references. So we have some regulatory books. We call them compendia, which you know about, or we call them pharmacopoeias, for example. They contain the you can say standards or standards of properties that the excipients should have in order to be accepted to be used in pharmaceutical manufacturing. We should be able to describe tests used for physical and chemical characterization of excipients, describe the impact of changes in excipient characteristics on the formulation performance. So here we should understand how important is the excipient to be standardized for the physical and chemical properties to be determined, so that because it may affect the performance of the formulation. And we should be able to list the conditions that must be met by an excipient to be accepted by regulatory bodies for use in pharmaceutical formulation. When we say regulatory bodies here, we mean the authority in each country that is responsible for registration of pharmaceutical products. Like, for example, in Malaysia, we have NPRA. Like America, we have the FDA. So this is this body we call as, or this authority we call as regulatory body. Let's have some introduction first, and before we start talking, look at these tablets. I try to find different some pictures that may give you some clear picture in your mind when I talk about excipients. So this picture attracted me actually. Usually in tablets, you will not see this kind of different colors, right? Mostly tablets will have one uniform color. But I choose this one specifically so that because it tells you that this tablet looks like having different ingredients. That's why you have some different colors in the tablet matrix, right? So that's the situation actually. Although in many formulations may not be visible, most of the ingredients may have the same color, white or off-white, but most of pharmaceutical formulations will have several ingredients. Maybe one or two of them only are active ingredients, the rest are called excipients. So drug substances, the drug substance itself that has pharmacological effect, we call it active pharmaceutical ingredients. So these drug substances or active pharmaceutical ingredients are usually given as a part of a formulation. When we give you a tablet or ampoule containing solution or syrup as a liquid, it's not only composed of the drug substance, but the drug substance is just a part of the formulation. There is recipe that was made to make this formulation. So this API is in combination with one or more non-medicinal agents that we call excipients. So the substance that have pharmacological effect, we call it drug substance or active pharmaceutical ingredient. Anything else in the formulation, we call it as excipients. And these excipients serve varied and specialized pharmaceutical functions. So it is intended to be included in the formulation, not for pharmacological function, not to give pharmacological effect. It should be pharmacologically inert, but it provides pharmaceutical function that we'll explain later. In the dosage form, the total amount of excipients usually or frequently used is greater than the amount of active drug substance. You may hold in your hand a tablet that weighs about 300 milligrams, 300 milligrams tablet. The API or drug substance is only about 50 or 60 milligrams. The remaining, so it's about only 20%. The 80%, the remaining 80% of that tablet is all excipients. Okay. Sometimes 50-50, sometimes drug substance has a larger percentage compared to the excipients, but in most cases, excipients are greater in amount compared to the active pharmaceutical ingredient. So we need to have a proper consideration of the physical, chemical, and biological characteristics of the APIs and excipients used in the formulation, as well as their compatibility with one another. And this is essential to produce a stable and effective drug product. So if you want to have a stable drug product where there will be no accelerated degradation of the API or no interaction with the other components, then we need to choose excipients that are compatible. Compatible simply means they don't interact. They don't have a chemical reaction with our drug substance so that the drug substance will remain stable. So during the shelf life, when you take the product before the expiry date, it will give a good pharmacological effect. Okay, we need to look for the physical characteristics, chemical characteristics, and biological characteristics. For example, you need an excipient to make a sterile dosage form. For example, like ampoule for injection, okay, solution for injection packed in ampoules. Or you want to use excipients to prepare some powder for injection, for example, that will be dissolved and then injected. So all the components must be sterile. When we talk about biological characteristics here, we talk about sterility. But if you use excipients to make tablets or syrup, for example, you don't need the excipients to be sterile. So you have to consider, depends on the dosage form we're preparing. We need to use excipients of appropriate characteristics, biological characteristics, physical and chemical. Physical characteristics, then we talk about, for example, particle size, viscosity, and so on. When you talk about chemical, then we talk about the assay, the purity, the limit for, say, heavy metals, limit for impurities, and so on. So now we talk about the rationale for inclusion of drugs in dosage forms. Why do you need to include or to prepare the drugs in different dosage forms? When we say dosage forms here, means we means by it different forms like pharmaceutical forms like tablet, capsule, in solution for injection, ointment, cream, and so on. They are all considered or called dosage forms. Okay, so usually we make the drug substance into a form that is suitable to receive the appropriate dose of the drug. Suitable to receive the appropriate dose of the drug. So why do we need to do that? Why do we need to prepare pharmaceutical dosage forms, not to just give the active pharmaceutical ingredient or the drug substance pure? That is for safe and convenient delivery of accurate dosage of drugs. Especially potent ones, which are administered in milligram or less than milligram doses. Some drugs are even administered in microgram doses. How, as a patient, if I give you 20 milligram, that is enough to consume, for example, say for 1 month, how will you be able to weigh 125 micrograms per dose? How you are going to administer? So I must make it in a form that you can hold with your hand or measure with a spoon or take into a syringe and measure in the syringe, determine the volume that you withdraw with the syringe, how much is it that you need to inject, so that you can take accurate dose of the drug. The dosage form can also include excipients that serve specific functions. So I make it in a form, and this form usually needs the addition of excipients to serve specific functions. Now we talk about the functions of excipients. What are the functions that excipients will perform? First, they can protect the drug substance from destructive influences of many factors like atmospheric oxygen or humidity. So if some drugs can be easily oxidized, and then means that they will be degraded, they will not be stable during that storage. I can add some excipients like antioxidants that will protect them from oxidation. I can, this one for protection from humidity, I can coat the tablets with substances that prevent the permeation of moisture. From oxygen also, I can, when we prepare ampoules, then we can seal the ampoules, and then we can have an inert gas like nitrogen or carbon dioxide, like nitrogen, for example, or carbon dioxide uh, in the space above the liquid level. We can use some excipients to protect the drugs from gastric juices. Some medications that may be destroyed by gastric juices are prepared as tablets. I can coat the tablet with a material that doesn't dissolve in the acidic pH of the stomach. We call this form as enteric coated tablets. And these tablets, after swallowing, passing to the stomach at acidic pH, the coat will not dissolve. The tablet still remains intact. And then after it passes to the duodenum and then jejunum, where the pH will be neutral and then turning to alkaline, then here only the coat of the tablet will dissolve, and then the tablet will disintegrate and the active ingredient will be released. And in that alkaline pH, it will not be destroyed. So this is one way to protect uh, drug substances that are destructed or degraded by acids. Can be also used to mask an unpleasant taste or odor. So, for example, if you have a drug substance with a bad taste, you can prepare uh, in dosage forms like capsules. So the capsule shell itself is a barrier between the mouth or tongue and the powder, so you will not taste it when you swallow it. Or you can make some sugar coating, for example. Use coated tablets with different types of coats, including sugar coat. So instead of tasting the bitterness of the drug substance, you will taste the sweetness of the sugar coat of the tablet. And then once you swallow the tablet, then the sugar coat will dissolve. The tablet will be exposed to the gastric juices, will disintegrate. The drug substance will be released. And you can also use flavored syrup. And a long time ago, medicine, especially syrups, was very bad experience for children. Nowadays, once the children taste the medicine, they want more. Why? Because of the flavor of the syrup. We have now syrups with different flavors. Tastes fruity and very sweet and very nice. So the children will not run from taking the medicine. Also, excipients can provide a dosage form suitable for application into specific site. For example, you want to insert the drug substance into body space or compartment. For example, we want to give into the rectum, then we need hard, for example, dosage form like having the shape of suppository, for example. So we need to mix the drug with materials that are solid, and we put it in a mold and give it the shape we need so that it can be inserted into the rectum. Same for the vaginal suppositories for vaginal infections of women. We may need to give it by injection, so we need to prepare a solution, for example. So we prepare solution, then we need to use some excipients, like at least solvents, to dissolve the active ingredient, make solution out of it. And sometimes we need to use to prepare to generate aerosol to be inhaled into the lungs. So in this case, we will mix the drug substance with excipients like gases, compressed gas, that will be liquefied. And then once you actuate the the, the, this inhaler, it will generate aerosol that can be inhaled into the lung. For oral administration, I can add excipients to prepare liquid or solid dosage forms, like tablets, capsules, and syrups, for example. So in syrups, I will use sugar substance. I will use water as a solvent. I may add some preservative, whatever. And then for tablets, I need to add many ingredients that I'll mention later. What's the function of the tablet? Um, I can also use some excipients for preparation of liquid or semi-solid dosage forms for topical administration to prepare lotions, creams, ointments, and transdermal patches. Lotions may be in the form of emulsion, maybe hydroalcoholic. Creams are usually, and ointments will have some fatty substances. Creams will have also, in addition to the lipid or fatty substances, I need to add surfactants so that or emulsifiers to make emulsion. Creams are usually emulsions, either water and oil or oil and water. Most of them are oil and water emulsions. Sometimes I need to the drug to have prolonged effect. So I prepare some special dosage forms that don't release the drug immediately, but release the drug slowly. Example are transdermal patches, some injections that have long release. For example, you have penicillin. Some types of penicillin, for example, are given every 1 month only, and then we have controlled release tablets. So for such dosage forms, there are special excipients that I add, and they have some mechanism to control the release of the drug substance from the tablet or from the suspension or the injection, whatever the dosage form we prepare. So here, let's go back to I think now you imagine what's the meaning of dosage form? So, or drug product. Drug product is a finished dosage form, capsule, solution, or tablet, for example, ointment, cream, syrup, and so on, containing two things. It is composed usually of a mixture of the drug substance or active pharmaceutical ingredient and the excipients. Can be excipient, can be excipients. Okay. So it contains active drug ingredient in association with the excipients that make up vehicle or formulation matrix. So excipients will make the formulation matrix. API, this is the material that have the pharmacological effect. So it is the medicine itself, the ingredient or ingredients of a pharmaceutical product responsible for its pharmacological activity. We can also call it medicament, we can call it drug substance, we can call it active ingredient. Anything else in the formulation is an excipient. So the drug substance is API or medicament or active ingredient or active pharmaceutical ingredient. Anything else we call it as excipient. We call it excipient. So excipients are pharmaceutical excipients are substances other than the active pharmaceutical ingredient that have been appropriately evaluated for safety and are intentionally included in a drug delivery system. So let's talk here about the word intentionally. So have been appropriately evaluated for safety, so they must be safe, and are intentionally included in drug delivery systems. So I put them for a purpose, to serve certain function. We will mention more functions later on, functional categories. Okay, so are the excipients regulated or no? Excipients are regulated. Why they are regulated now? Regulated means that there are some standards has been set up by regulatory bodies, drug registration authorities, that excipients must fulfill. Otherwise, they will not be allowed to be used in pharmaceutical formulations. Okay, this is the meaning of the regulation. Means that we have some standards and acceptance criteria. If the excipient doesn't fulfill this criteria, then cannot be used. Why there was some regulations regarding excipients and why they are growing? Usually, of course, after some fatal mistake or some disaster, then a swift action must be taken. So it happened before that there was some sulfa drugs, sulfanilamide was prepared as elixir in USA. And they used diethylene glycol as solvent, or co-solvent here, in this formulation, and it resulted in death of 107 people, okay, due to the toxicity of this solvent, diethylene glycol. So when this accident happened, of course, the media will talk about it. A lot of noise will be there in the society, a lot of demands, how this can happen, why the government doesn't control the industry, why they don't have regulations to prevent such mishaps from happening, and so on. So then this incident dramatized the need to establish drug safety before marketing. So now they understood that there must be enough studies to be performed and to prove the safety of the formulation before it is allowed to enter into the market. And this provided an impetus to pass the pending Federal Food, Drug, and Cosmetic Act of 1938. So the act was there, prepared, not yet approved. Maybe nobody um, sees that there is urgency to approve it. Maybe still so many arguments about it and so on. But when this happened, then it accelerated the issuance of this act, FDA Act, Food, Drug, and Cosmetic Act. So in these regulatory guidelines, these are the conditions that have been stipulated regarding the excipients used in the pharmaceutical formulations. Number one, the excipient should has no harmful or toxicological effect. It should have good stability and should has no compatibility with the drug. There should be no drug excipient incompatibility. Means that they don't interact physically or chemically in a way that will deactivate the drug substance. And this interaction also should not happen by any impurity or in the excipient. There should be no interference in quality validation analytical tests. That means that when you perform a chemical test, for example, like assay, if we perform any other test to test the quality of the medical product, the excipient should be chemically inert or should not interfere with that tests and should not give false results. Should not give false results. Okay? So when you conduct a test, when you carry out a test to verify the quality of the product, then the excipient should not interfere with the test and results in false results. And it should has no problem or no interaction with the primary packaging materials. We have primary and secondary packaging materials and even tertiary. Primary packaging material is the packaging material that comes in contact with the product itself. So for example, you can see, you may see an ointment or cream in a tube, and this tube is in a box, carton box. So the tube itself, the inner wall that comes in contact with the ointment or cream is the primary packaging material. And then anything else is secondary packaging material. For example, the carton box is called secondary packaging material. It should be physiologically inert, means it has no pharmacological effects. It should has no unacceptable microbiological burden. So if we need to use it for preparation of sterile preparations, it should be sterile. If we need to use it for preparation of non-sterile preparations, then it should not have a microbial count exceeding the allowed limit. Satisfaction of regulatory issues and guidelines in all countries where the product is to be marketed. So now we have different regulatory bodies, we may have different compendia. You want to export your product to USA, then you have to follow the FDA regulations and USP, United States Pharmacopoeia. United States Pharmacopoeia. If you want to export your drug to India, then you have to follow the Indian Pharmacopoeia. Japan, Japanese Pharmacopoeia. European Union, then European Pharmacopoeia, and so on. So it should satisfy the regulatory issues and guidelines in the countries where the product is to be marketed. Should be satisfactory for environmental issues, not harmful for the environment, and so on. When it comes to degradation after disposal, will degrade in the environment, will not form toxic degradation products, and so on. And then ease of accessibility, distribution, and economical cost. We want always to use excipients that are available. There is no shortage in the market, and preferably I have suppliers in my country. If not, then should be nearby where there is logistics matters are easy and at a good price so that my product will not be expensive at the end. Usually excipients may have different names. So for the same excipient, I may have several names. So for example, one polymer used, we will mention later on in the functional categories, used for different functions. In general, it has a high viscosity, can be used for modified release tablets, as a stabilizer and emulsions, can be used for many purposes due to this high viscosity. You can check one textbook, one monograph. You will find that the name, for example, is hypromellose, methyls, carboxymethylcellulose, hydroxypropylmethylcellulose, HPMC. You can find also other names for it. So hypromellose, HPMC, maybe other names. So now if we want to write the label of our product, we want to write the ingredients, active and inactive ingredients. This is the standard now in many markets that in the label you will find that they declare both active and inactive ingredients. For example, you want to take paracetamol tablets, they will not mention that it only contains paracetamol, but they mention paracetamol and other components as well, the excipients besides the drug substance. So which name we are going to use in our label? So it should be taken from the current edition. The name should be taken from current edition of one of the following references or reference works in that order. So first I look at the USP or national formula. If I find it there, there is monograph right in USP. It is mentioned in USP and there are characteristics mentioned there where the standards are given for the substance. Then I use the USP name. So which USP name? The name used for the title of the monograph. Used in the title of monograph. So if the title of the monograph is hypromellose, I write hypromellose. If the title of the monograph is hydroxypropylmethylcellulose, then I use hydroxypropylmethylcellulose. Okay. How about if no monograph in the USP? Then I should look in USAN and USP directory, a dictionary of drug names. Okay. If not, then I look at this reference cosmetic ingredient dictionary. And then if not, then I look for food chemicals codex. Okay. So as long as I found in one of them, I should not look for the others. I just use the name in the first one I find the name within. Now let's have some round. We go around and see different functions that excipients can do, can perform, and then it can perform in the pharmaceutical formulation. And we see examples also about excipients used for such functions. And this actually is this functional category is a qualitative classification. We just say it is used for some purpose. We don't quantify anything. So this describes the purpose or the role of an excipient in a drug product. So number one, we have some excipients used as acidifying agents, and these are used in liquid preparations to provide acidic medium for drug product. I can also use alkalinizing agents to give alkaline pH to the drug product. So why do I need to do acid to make the pH of the product acidic or alkaline? We need, for example, to use a pH where the drug is soluble or the drug is stable. Usually, I need to consider two things for choosing the appropriate pH of the material, or even three things. where I will apply it, for example, I apply it to the eye, so it should not be very far from the pH of the eye, so that will not cause, for example, stinging or irritation. Uh, and then second thing is solubility. I want to dissolve my drug, so some drugs may be soluble at acidic, some drugs are soluble at alkaline pH. That's why I need to control the pH of the formulation. Especially here, we talk about liquid formulations. And then sometimes also I need to choose the pH near to that will cause the drug to dissolve, but same time will not cause degradation of the drug. So for acidifying, I can use acids like citric, acetic, fumaric, hydrochloric, and nitric acid. For alkalizing, I can use alkaline substances like ammonia solution, ammonium carbonate, mono and diethanolamine, potassium and sodium hydroxide, sodium bicarbonate, sodium borate, and sodium carbonate. And I can use toluene. These are all alkalizing agents. I may need in some formulations to add adsorbent, and this is capable of holding other molecules onto its surface by physical or chemical chemisorption. By physical or chemical means, okay, chemical means we call it chemisorption. So this adsorbent will absorb uh, probably the drug substance on its surface, like powdered cellulose or activated charcoal. Air displacement for drugs that may be, for example, oxidized by air, by the oxygen in the air, then I need to replace, as I mentioned before, in the ampoule, for example, the air above the liquid surface with inert gas like nitrogen or carbon dioxide. So this agent is employed to displace air in the hermetically sealed container to enhance product stability. Hermetically sealed is the ampoule usually is closed or sealed by heat. It will melt and close. So this is what we call hermetically sealed. If I want to prepare aerosol to be inhaled into the lung or even to spray on the skin, like what you see in the football matches, when the player will be injured and then they spray something, this is uh, aerosol generated. And inside the canister, usually we have propellant. Propellant is the inert gas that is compressed into a liquid. These are agents responsible for developing the pressure within an aerosol container and expelling the product when the valve is opened. Examples are carbon dioxide and the fluorocarbons, different fluorocarbons like dichloro, difluoromethane, dichlorotetrafluoroethane, trichloromonofluoromethane. In formulations, especially the aqueous formulations, if there is water, then it is a medium for bacteria and molds to grow. So I need to use, in this case, I need to use preservatives. If I have an oil medium or solid dosage form like tablet, I don't need usually to add preservatives because protected from moisture, then no fungal or bacterial growth will happen in that dry environment. So usually in aqueous liquid preparations, I need to add preservatives like the parabens, methylparaben, ethylparaben, propylparaben, butylparaben, and so on, and then antimicrobial like benzalkonium chloride as well. In some formulations, if my drug is sensitive to oxygen and can be easily oxidized, then I need to add antioxidants. So it all depends on my drug. I don't need to have antioxidant in all formulations. And this prevents the deterioration of preparations by oxidation. And these are examples of antioxidants like ascorbic acid, butylated hydroxyanisole, butylated hydroxytoluene, propylgallate, and so on. But simply, I can tell you that we classify them usually into two classes based on their solubility. Some of them are lipophilic, some of them are hydrophilic. So in case we have an aqueous formulation, then we use a hydrophilic antioxidant. If we have a lipophilic formulation like ointment, for example, then I should need a lipophilic antioxidant. I can use some buffering agents to control the pH of the medium, which is usually salt plus acid and its salt, or alkaline and its salt, as you studied before in physical pharmacy or even in chemistry. So I can use like potassium metaphosphate, potassium phosphate, more basic sodium acetate. sodium citrate, anhydrous dihydrate. Usually I add some of the salt and then I adjust the pH with the acid. So I'll have a mixture of acid and the salt. In some formulations, I use chelating agents. Some people, they can be pronounced also as chelating agents. Both are correct. You can say chelating or chelating agent. These substances usually will bind with metals, with heavy metals. They form stable water soluble complex or chelates with metals. I use them in some liquid pharmaceuticals as stabilizers because these metals may promote the degradation of the active ingredients. So I want to remove them from the medium by addition of chelating agents. They will interact them and they will prevent them from interaction with the active ingredient. The example is EDTA and disodium EDTA. Some formulations I may need to add colorant to give a color to the liquid or even to the solid, sometimes to the coat of the solid dosage form, like tablets. The tablet may be normally white, and then the coating material, I put some colors. Capsules also usually colored, okay? We may need to add coloring agents just for cosmetic effect or for protective effect as well from light. So you have different uh, types of coloring agents. Each of them has different color. Some are red, some are yellow, blue, green, orange, red, and so on. Ferric oxide can be used to give a red color, caramel also to give the color of caramel. Some excipients used in some formulations are clarifying agents. They are used as a filtering aid because of its adsorbent qualities like bentonite. Emulsifying agents, they are used for making emulsions and like creams and lotions. They promote and maintain dispersion of uh, oil in water or water in oil to make an emulsion or cream. Examples are acacia, cetamacrogel, cetyl alcohol, glyceryl monostearate, sorbitan, polyoxyethylene 50 stearate. Encapsulating agents used to form thin shells to enclose a drug for ease of administration, like gelatin. So gelatin usually is the material that we use to make the capsules. Okay, so the hard capsules or even soft capsules that contain liquid inside is made of gelatin. Gelatin here is the encapsulating agent. And it's also used for microencapsulation. It's used also for um, nanoparticles making as well. So it's an encapsulating agent. Flavorants, you can add flavorants to impart pleasant flavor and often odor to the preparation. So it will give a pleasant taste. We have anise oil, cinnamon oil, cocoa, menthol, orange oil, peppermint oil, and vanillin. They can give different flavors. Humectant is a material that will keep the moisture. They will keep the moisture, used to prevent drying of the preparation, particularly ointments and creams. So, especially creams, I don't want them to dry or ointments upon storage. So, in the matrix, I can add some humectants like glycerin, propylene glycol, and sorbitol, and they will keep the moisture so the cream will not dry up upon storage. Some are used as levigating agents like mineral oil, glycerin, or propylene glycol. Um, I use them to when I want to mix at the beginning, when I put the active ingredient or other substances that have large particle size. I add the oil and then I grind them. They will become very fine in the mortar. Then I can add the other components of the formulation. Some excipients are used as ointment base. When I want to make ointment, then I mix the drug substance with fatty materials like lanolin, like hydrophilic ointment, polyethylene glycol, petrolatum, white ointment, yellow ointment, rose water ointment. And this will be the vehicle that will carry the active ingredient. Some excipients are used as plasticizers. So if I want to coat my tablets, for example, if I want to coat the tablets with some coating material, this material may be brittle, may be easily broken. if used alone to make the coat. So in the solution I prepare where I dissolve the coating material, I add plasticizer with the coating material. This plasticizer will make the film more malleable, enhance spread of cover over tablets, will become uniform cover, uniform thickness, whether I cover tablets or beads or granules, and it will not be broken on upon storage or upon processing like packaging and so on. Example, diethyl phthalate and glycerin. They are plasticizers. If added to the coating solution that's sprayed on the tablets to make the coats, then they will make it more stable. And of course, solvents is a very important category. Many materials are used as solvents. Some are aqueous, some are non-aqueous materials, and they used to make liquid formulations. They used to dissolve another substance in preparation of a solution, maybe aqueous or not. Some will be used as co-solvents if the drug doesn't dissolve in water alone. So I make a mixture of water and this substance, this solvent like alcohol or glycerine or propylene glycol, for example, so that it will help to dissolve the substance that doesn't dissolve normally in water. And if I'm preparing injections, then the solvents must be sterile. And these are examples like I can use alcohol, I can use different types of oils like corn oil, cottonseed oil, peanut oil. They are used actually to make the solution for injection. The one that is for long, for prolonged effect. Glycerin and isopropyl alcohol, they are used as co-solvents usually with water, and alcohol also can be used as co-solvent. So I make mixture of water and alcohol, water and glycerin, water and isopropyl. Isopropyl alcohol is not for internal use, for external use only. Some mineral oils may be used as solvents, oleic acid, purified water, sterile water for injection, sterile water for irrigation. They are all used to make solutions. Okay. Some materials are used as stiffening agents to increase the thickness or hardness of a preparation, usually ointment or cream. So, for example, you make emulsion, normal emulsion, just use emulsifier, water, emulsifying agent, water, and oil. Then you get something like the milk, just a liquid like milk. So, how about if I want to make it little thicker? Then I can add stiffening agent, then I can make a lotion, like Nivea lotion. Okay? You can add more, then you can make it semi-solid like ointment, like cream, for example, like cream. Ointments also, I can use stiffening agents to control the viscosity or consistency of the ointment. I want it harder, I want it softer, and so on. I can add these kind of waxy materials to also control the consistency. So examples of stiffening agents are cetyl alcohol, stearyl alcohol, microcrystalline wax, white wax, yellow wax, cetyl esters wax, and paraffin. Okay. Suppository base. Some excipients are used for making suppository, like cocoa butter, for example, the most famous example. I mix the drug substance with cocoa butter. I put in a mold to give the shape of the suppository. Then I made the suppository like that. So they are vehicle. Same like the solvent in the liquid formulations. Cocoa butter here is the vehicle that carry that within its matrix the drug substance will be distributed or dissolved. Can be dispersed, can be dissolved. I use also some other materials like polyethylene glycols can be used for making PEGs, polyethylene glycol. They have different grades of hardness and consistency. They are also used to make suppositories. Some materials are used as surfactants. You know how they work from physical pharmacy already. And they are used as wetting agents, detergents, or emulsifying agents. And we have examples. Suspending agents, they are materials that usually have some kind of viscosity. So if I add them to they are viscosity increasing agents. When I add them to the solvent, then they make it thicker. So once you shake and you disperse the suspended particles, they will not sediment quickly. They will not precipitate quickly. It will take a time to precipitate. So it will give you the time as a patient after you shake the bottle, then the dispersed drug substance will be homogeneously dispersed for some enough time till you take your dosage, till you take the required dose. Then in this case, we have a homogeneous distribution, so you will take the accurate dose. And these are examples. Some of them are polymers like carbomar, agar, tragacan, carboxymethyl cellulose, hydroxyethyl cellulose, and so on. Okay, hydroxypropyl cellulose, hydroxypropyl methyl cellulose. This is HPMC I was mentioning, and kaolin and kaolin is metallic, inorganic. Okay, some are used. Some excipients are used as sweetening agents. I make a serum, for example. I want to make it sweet, so I can add sweetening agent to impart sweetness to the preparation, like aspartame. This is synthetic. Dextrose is natural. Glycerin natural. Mannitol is natural. Saccharin sodium is synthetic. Sorbitol is natural, and sucrose is natural. Some materials are used as tablet anti-adherent. We want to prevent tablet ingredients from sticking to the punches and dyes during the production. So in the tablet press machine that will compress the powder into tablets. In the metal parts that compress, we have two sides usually, and the tablet will be compressed between these two metal parts. The powder will be compressed between these two metal parts to make a tablet. So for the ease of production, I want the tablet to be formed and then ejected easily to be collected in the vessel. So usually I need to add some fatty materials like lubricant. It has lubricant effect, so it will prevent the adherence between the powder and the metal parts of the instrument, which are punches and dies. So it can be ejected and released from the machine easily and collected in the vessel. Example, magnesium stearate. For tablet making also, some of the excipients used are tablet binders. These are substances used to cause adhesion of powder particles in tablet granulation, so that the tablet will not be friable and will not be broken easily upon shipment. So we use tablet materials that act as tablet binders. They bind the particles together. They stick together in a solid matrix. And some of them are like acacia. Acacia is the Arabic gum. It's a gummy material. And some are alginic acid. All these materials are called sticky materials. Carboxymethyl cellulose as well. Cellulose ester, cellulose polymers as well. They are like gum or like glue, and they are viscous at the same time. They have different grades, and these grades of viscosity are suitable for different applications. When we want, for example, sustained release, we use high viscosity grades. When we need just to be binder or to be, for example, stabilizing agent and suspensions, we can use lower viscosity grades. Okay, so some of them are natural, like starch, and some of them are synthetic, like povidone and carboxymethyl cellulose is semi-synthetic because it is derived from cellulose that is natural. So some of them are natural, some of them are synthetic. Still, we have a lot to go through. Okay, we need to have a break for 5 minutes and then we can continue. Another functional category is tablet and capsule diluent, and this is an inert filler to create desired bulk, flow properties, and compression characteristics of tablets and capsules. What does this mean? Means I add it to the drug substance when I want to make tablet. To create the desired bulk means to make the tablet weight to the required weight. I have only 5 milligram of active ingredient, for example, which is very tiny, cannot make a tablet. I want to take a tablet that weigh about 100 milligrams. So I need to add about 95 milligram of another substance to make up the total weight. So it makes the desired bulk. Second thing, it makes the flow properties. For the purpose of manufacturing, I need the powder to have a good flow so that will flow regularly in the tablet machine so that I will have uniform tablets of the same weight. So I should choose also diluent that have good flow properties, can improve the flow properties of the whole mixture. And then also to provide compression characteristics means that should have good compressibility. So when the tablet machine compress the powder, it will be compressed into a solid tablet and will not have any problem in compression. Sometimes when we want to develop or we make different types of or different formulas and then we test them. Some powder mixtures may not be compressed easily. I use high compression force, but still I don't get a tablet. I get just only fragments, okay, that can separate from each other. So this is what we mean by compression. Should have good flow properties, compressibility, and then will make up the weight of the tablet. And these materials all should be inert materials as well, don't have pharmacological effect. And several materials can be used as diluent. Some of them are cellulose and cellulose derivatives. Some of them are mineral, like kaolin, dibasic calcium phosphate, and precipitated calcium carbonate. Some of them organic, like sorbitol, starch, and so on. Okay, these all substances can be used um, in the mixture. Based on what I choose them, as I mentioned before, number one, they should not interact with the active ingredients, so no incompatibility with the active ingredients. Second thing, they should have a good flow properties and compression characteristics as well. Okay. Um, when we make tablets, usually we make a paste, then we turn this paste into granules, and then we dry the granules because granules has better flow compared to the fine powder. If I didn't make this process of granulation and drying of to dry granules, then compression, if I directly mix the powder ingredients and compress them, then I call it direct compression excipient. So some excipients are used as tablet direct compression excipients. So used in direct compression tablet formulations means I don't need to do granulation. And actually, from some of these materials, I may have like mannitol. I have fine powder that used for conventional tableting process through granulation first. And I have also spray dried lactose, I mean lactose, and then I have spray dried lactose. Um, this has a more like granular. They have a larger particle size, have better flow properties. So if I want to, depends on whether I want to make my tablet by direct compression or I make granulation first. If I want to granulation first, I can buy the fine lactose, fine powder. If I want to use for direct compression, then I have to buy the direct compression grade of lactose. Same goes for others, mannitol, microcrystalline cellulose, and so on. So they can be the company itself that produce these ingredients. They produce them in two grades, normal grade and direct compression grade. What's the difference between them? Direct compression grade is having larger particle size. What's the benefit of larger particle size? They have a better flow properties. So I don't need to do granulation. They are already like small granules. Tablet may contain, what did we mention up till now? We mentioned for tablet making, the tablet formula will have anti-adherent, will have binder, and then we'll have diluent. Okay, diluent is the major component beside the drug substance. And then it may have small amounts of other things like anti-adherent, binder, and disintegrant. We said anti-adherent will prevent sticking to the punch and die, to the machine. Binders will bind the particles or granules together so that we can get a solid tablet. Disintegrant will work after administration of the tablet. The tablet is solid, but I want it to break up easily and quickly once swallowed and reach to the stomach, for example. So there must be a substance that will absorb water, swell, and blow up the tablet from inside. So this is the function of the disintegrant. It's used in solid form to promote disruption of the mass of the tablet into smaller particles more readily dispersed and dissolved. Where does this happen? Happens after swallowing the tablet when in contact with the gastric fluids. These are examples like alginic acid, uh, cholecryline, potassium, sodium alginate, sodium starch cholecrate, and starch. So how do they work? By absorbing water, and then they swell, and then they make an internal pressure inside the tablet that will blow it up. Okay, will break it into small particles that will disperse in the gastric fluid and then will dissolve and release the active ingredient. Some tablets may be coated, some may not. Many tablets are uncoated. In case we need to prepare a coated tablet, then we use coating agents to coat the tablets. We have different types of coats. We have sugar coating, film coating, enteric coating. Sugar coating, we use sweet substance like liquid glucose or sucrose in order to mask the taste. Film coating like hydroxy ethyl cellulose, hydroxypropyl cellulose, HPMC, hydroxypropyl cellulose, and so on. They are used to protect from moisture, from light, and so on. Okay, especially protection from light. I will add some coloring agent also as well. Enteric coating, we mentioned before that these substances will not dissolve in the acidic gastric fluid, but will dissolve in the neutral to alkaline pH in the duodenum to jejun. So in this case, the tablet will stay intact in the stomach, and the drug substance will be only released in the small intestine. Okay. Also use tablet glidant. This is another ingredient it's used. So see how many ingredient now is used in tablet making. These are all excipients to provide different functions. The glidant here will improve the flow properties of the powder mixture. So they help also for granules to slide uh, and not stick to each other. Okay, we use things like colloidal silica, cornstarch, and talc as glidants. We mentioned the lubricant before, which is anti-adherent as well. They are stearates or oils. And then tablet or capsule opacant. If I want to use a substance that will make the coating dark and prevent the light, then I used something like titanium oxide. It will be included in the coating solution. So the coating solution will have coating. These are the components will be coating solution. They have diluent or vehicle or solvent. I dissolve the coating material, and then I dissolve plasticizer in the same solution, and then also titanium dioxide will be added to the solution. Then this will be sprayed, and then we have a pan with hot air flow that will dry up the solvent and only leave the coating material on the surface of the tablets. And then I want this coat to have a shiny appearance. Then after coating, I can use a polishing agent like carnauba wax or white wax. They impart attractive shine to coated tablets. In some formulations, I need especially injection solutions given for IV or, for example, ophthalmic solutions. Okay, so parenteral and ophthalmic solutions and irrigation fluids that I use to clear the wounds. I don't want to cause irritation. I need to make the osmotic pressure of the solution or the injection, whatever I want, or the ophthalmic eye drops. I want it to be similar to physiologic fluids. So I can use tonicity adjusting agent like sodium chloride. Viscosity increasing agents can be used also in several formulations, including, for example, to thicken topical creams, in ophthalmic solutions to enhance the contact time in the eye, in suspensions to reduce the sedimentation, we mentioned before, as a stabilizer. Okay, so I can use viscosity increasing agents, and we mentioned it before already. And some of them that are synthetic are available for cellulose are available in different grades with different viscosities. So I choose the grade suitable for my application. The last thing is the vehicle, and this is the carrying agent used in the formulation or used in formulating a variety of liquids for oral and parenteral administration. Generally, oral liquids are aqueous or hydroalcoholic. If aqueous, I call them usually syrups. They will contain also some sweetening agent or sugar material, and then hydroalcoholic means they have a mixture of water and alcohol. This usually is used for non-soluble, non-water soluble materials. This formulation is called, dosage form is called elixir. Solutions for intravenous use are usually aqueous, but if I give intramuscular injection that is especially for prolonged effect, usually the vehicle is oleaginous. I say vehicle, which is more general compared to solvent. If you say solvent means that you have a solution, but if you just say vehicle, means that in this liquid medium you may have the ingredients dissolved in the form of solution, or you may have them dispersed as a suspension. Okay, so vehicle is just a carrier. It may be solvent and it may be also a suspending medium. Okay, so we have some flavored and sweetened vehicles like acacia syrup, aromatic elixir. When you say elixir, means you have some amount of alcohol there. Cherry syrup, cocoa syrup. Okay, when you say syrup always, then we're talking about mostly a substance like sucrose in high concentration dissolved in water. Okay, to give the sweet taste and also give it a thick or uh, viscous consistency. Some vehicles are oleaginous, used for making injections like corn oil, mineral oil, peanut oil, sesame oil. And you should know that we cannot inject the oil injections by IV route, cannot be given in the vein intravenously. They can be given only intramuscularly. And I have some solvents or like sodium. Okay, can use also bacteriostatic sodium chloride injection. Can use the sterile water for injection. Okay, so you can use water or can use normal saline. Sodium chloride usually normal saline. I can add some ingredients to it and prepare injections. Now we go for the second part, which is excipients characterization. Characterization means to determine the physical, chemical, and microbiological characteristics. Means we test, and we report the results of the tests. They may be chemical. It may include chemical tests, physical tests, and microbiological tests. So if I say characterization means testing and determining the characteristics of my material. Why do we need to, why it is important to characterize the excipients? Because their characteristics will affect the formulation stability and performance. The drug substance can be susceptible to chemical and physical changes induced by the properties of the bulk excipients. And we usually in our manufacture, development in the dosage form development or the formulation development, we implement the quality by design, product development strategies, and this place an increased emphasis on detailed characterization of excipients to achieve robust formulations and processes. Okay, what does we mean by this? Then I skipped one diagram. We can go back to it. We have a dosage form we want to develop. Okay, we want to achieve some characteristics in it. So one of them is identity and appearance. I may use some coloring agents. Okay, in the solvent or the coloring in the tablet coat, whatever. I want to achieve patient compliance. So I can improve the compliance of the patient by adding flavoring or sweetening agent. So they will not feel the bitter taste of the active ingredient. I want to have a manufacturable or a dosage form with good manufacturability. So here I have to choose the appropriate components so that I can manufacture it easily. And then I need some processing aids like binders and glidants as we mentioned for manufacturing of tablets, lubricants, compression aids, suspending agents, chilling agents, emulsifiers as we use in this for example, suspending agents in case of this of suspensions. Gelling agents, I may use them for making gels. Emulsifiers, I use them for making emulsions, lotions, and creams. Okay, so I should use different ingredients to give different functions to the formulation. We call these ingredients as excipients. These excipients must have compatibility. They must be compatible with the drug. And then these are the characteristics I want for my dosage form. I want efficacy, I want safety, and I want quality. Okay, to be safe, effective, and have good quality, then I need to have a stable dosage form. For stability, I can add some chemical stabilizers like antioxidants and chelating agents that I mentioned before. This for chemical stability. For physical stability, for example, in suspensions, I can add viscosity enhancers and gelling agents to improve the say stability and then to increase the viscosity and then reduce the sedimentation rate of the suspended particles, and then prevent also that they make a solid cake that after I shake again, they will not disperse again. Okay. I may use preservatives to prevent microbial growth. So this is microbiological stability. I can adjust the pH by pH modifiers to achieve a pH in the liquid formulation that is suitable to dissolve the drug, suitable to keep the stability of the drug, reduce the degradation rate, and so on. And for modified release and absorption, I also add so many things. Okay. So now, what is quality by design? Quality by design, I determine the what do you call it? I have two variables, input and output variables. Output variables, the characteristics of the formulation itself. And then input variables is the characteristics of the substances used, excipients and drug substance. Maybe I can test, for example, effect of particle size of excipient, effect of amount of excipient, effect of viscosity grade of excipient, whatever, on say drug dissolution and other outcome characteristics. This is the quality by design. And then I find out the relationship between these input variables and the characteristics of the formulation. And then I determine what characteristics of excipients and drug substances are needed to produce a drug having the required quality. We will teach this in details in the next year, inshallah, in FCP or manufacturing pharmacy. Okay, this is about the quality by design. Quality by design means you make experimental design, you vary the characteristics of the excipients used and the ingredients in the formulation, and then so you vary qualitatively and quantitatively, and then you test the effect of this variation on the outcomes required on the characteristics of the formulation, and then you determine the appropriate amounts and characteristics of excipients that will give you the good performance of the formulation. We have different chemical classes of excipients. Some excipients are are inorganic, like iron oxide we mentioned before. We use as pigment to give red color. Calcium phosphate that we use as a filter. Some are small molecule organics, like mannitol that we use as diluent or sweetener. Sodium citrate used as that is used as alkalizing agent. And some are large molecules, polymeric, like hypromellose and starch. Hypromellose is hydroxypropylmethyl cellulose. So they range from simple, usually highly characterized organic or inorganic molecules to highly complex materials that are difficult to fully characterize. Um, excipient variability and compendial tests for characterization. One excipient may be available in several grades, as I mentioned to you before, and supplied by different manufacturers. As we mentioned, for examples before, the grades may be related to the particle size, for example, normal grade, fine powder, or direct compression grade. And we mentioned about the viscous materials that may be available in different viscosity grades. So this will be related to the size of the polymer itself. Larger molecule size or molecular weight, then we have a more viscous uh, grade. Okay, so you may have different grades when it comes to physical or chemical properties. They may have some large particle size or different molecular weights or different particle size. Different grades of the same excipient will vary in polymer molecular weight, degree of substitution, particle size distribution, and morphology as well. So sometimes degree of substitution, methoxylation, for example, propylation of the molecule uh, of the cellulose with different degrees of methoxylation or propylation, they result in different physical characteristics as well. Some side groups may result in hydrophilic polymer, some will result in hydrophobic polymer. Okay, like ethyl cellulose is usually hydrophobic, methyl cellulose is usually hydrophilic. Okay, different manufacturers will employ also different synthetic schemes or isolation techniques that can result in slight differences in physical properties. So they may produce the same substance, same grade, but however, they use different schemes for synthesis. So they may result, or even for isolation, they may result in slight differences in physical properties like the melt temperature, crystallinity, loss on drying, particle size, and so on. And even some chemical profile differences like trace impurities may differ from one supplier to another. This is also related to the quality by design thing. This is what I mentioned to you. We have critical quality attributes for the final drug product. I want dissolution, for example, in certain rate. I want hardness, certain value. I want friability, certain value, and so on. These are examples of characteristics of excipients that will result at the end to variation in the quality attributes uh, of the final product. So I mean to study the effect of characteristics, microscopic properties or microscopic properties of the excipients on the quality attributes of the final product in a design called experimental design. This concept is called um, quality by design. Okay. The result of the variation in properties may result from uh, different manufacturing. Uh, for example, batch versus continuous, different method of harvesting, extraction, different chemical synthesis scheme. You already studied medicinal chemistry, and then you show organic chemistry. Already you saw that I can design. I want to get from one starting material another, for example, alcohol or acid or organic alcohol. Then I'll design, maybe I need to do, for example, 2 or 3 reactions to get aldehyde, for example. I need to convert alkane to alcohol, alcohol to oxidize alcohol to aldehyde. Maybe you can have different schemes, okay? Or you can use different starting materials to reach to the aldehyde at the end. So based on the different schemes may have different properties. Um, Usually we have compendial. If you want to test or characterize your excipient, then I should refer to the compendia. Compendia is issued by regulatory bodies, and there is monograph for each substance. In this monograph, they state the standards, the characteristics required for the excipient. So formulation scientists rely on numerous compendial excipient characterization, chemical and physical uh, analysis, test methods, and develop novel methods to analyze key quality materials attributes of the formulation. USP general chapters, we have specific chapter monographs for substances, and we have general chapters as well in the USP. Please open the USP, go to the library, visit the library, find the USP we have, and open it and see the contents, see the different chapters there, see what formulation you can find in the USP. So We have some general chapters. They contain test methods that cover elements of chemical and physical properties at various levels, molecular level, for example, to determine the molecular structure, how to perform NMRIR, NIR, and UV, uh, nuclear magnetic resonance, infrared, near infrared, and ultraviolet spectrophotometry. And on particulates level, if I want to determine characteristics of the particles, like distribution of particle size and particle size, determination of particle size and particle size distribution. We say distribution here because I want to determine, we don't have uniform particle size in each granular material or granular material. So it's not enough to determine the average particle size, but also the distribution. How much percentage in what range? So usually I'll get a result of about maybe 10 bands. Each band is a range of particle size, and then I'll determine how much of the total mixture of particles is in this range, maybe 20% in one range. Another range I have 5%, another range I have 30% of the total mass in this particle size range, and so on. And then on the bulk level, I may determine the viscosity, loss on drying, thermal analysis, and so on. So there are different characterizations I can do for the excipients. And these methods provide core testing protocols for routine certification of materials for release specification, certificate of analysis, and compendia compliance. Okay, let's explain this. Number one, to use the material, it must comply with the compendial requirement. So for example, FDA, they issue USP, and then USP is the reference based on which I can decide whether the material should be accepted or uh, rejected. So the say for example lactose, I will find lactose monograph in the USP, United States Pharmacopoeia. I should open it. I will find a list of tests that should be performed on this lactose and the expected results or the accepted results. Okay. Then I should prepare something called certificate of analysis. Or before certificate of analysis is okay. Then I should prepare some certificate if I receive it in the test. Okay. Number one, I should prepare the release specifications. So I should have a document that contain this list of tests and accepted results. Okay. When you receive the material from the supplier, you cannot use it directly in the company, but you need to test first, and then based on the test results, if they fall within these ranges, accepted results or specifications, then I can use it. If not, then I should reject. I should return it back to the supplier. And the results of the tests will be recorded in a certificate called certificate of analysis. It will contain the same list of tests, also another column for the accepted results or the accepted or for the specifications, but we'll have additional column for the results. If the result is within the accepted range, then it passed the test. Okay, so the decision that this substance is complying to or having the required characteristics will be based on the compendia like USP or European Pharmacopoeia. It will contain a list of tests that should be done to this material, physical, chemical, and microbiological. I should transfer the tests mentioned in the monograph to some document that will contain the release specifications of the material, list of tests and accepted results. And then any material, once I receive any amount of this material to my company, to the factory, then should be tested, and then a certificate of analysis is issued. It will contain the results of the tests and final decision. If all the results are within the specified ranges or specifications, comply with the specification, then can be accepted, pass, and then can be used in the company. Otherwise, should be rejected and returned to the supplier. The objective is to identify excipients and ensure quality through routine analysis. So these tests are usually including identification test. Number one, I should make a test to ensure that the material I receive is the material that I order. I want lactose, then I should do tests to ensure that this is lactose. Second thing, I want to do some other tests to ensure the quality, maybe assay impurities, physical tests, for example, to ensure that the particle size is within the range I want, whatever. And metal, for example, maybe have a limit test for heavy metals, should not contain cadmium or other toxic heavy metals, for example, should not exceed certain range and so on. Okay. The tests are usually done to the materials are divided. You can classify them into universal tests and specific tests. Universal tests are being done to all substances listed in the pharmacopoeia, and they evaluate general characteristics like identity, purity, and safety. Purity is the assay, and we do active ingredients and so on. We do impurities as well, because some impurities are toxic, so this is for safety. Examples are identification, assay, pH, loss on drying, heavy metals, microbial limits. And the focus here is general quality test assurance and applied broadly across different substances or across different substance types. So I do assay identification, pH, loss on drying, and so on for most of for APIs, excipients, and dosage forms as well. Okay, so you will find monograph for example, paracetamol as a raw material, as a as a medicament for drug substance. And you have other monographs for paracetamol tablet, another monograph for paracetamol syrup, and so on. This is the dosage form. Okay. Specific tests, we usually apply them to specific substances or formulations with unique properties to evaluate specific characteristics critical to the substance function or intended use. Example is dissolution, friability, sterility, uniformity, content uniformity, endotoxins, and so on. Here, the example is they don't apply for everything. For example, I do endotoxins only if the preparation or if the material is to produce a sterile dosage form, or if the dosage form is a sterile dosage form. For example, if I want, I have, say, antibiotic powder, uh, the drug substance itself, as intended to produce a sterile injection, antibiotic for injection. Then endotoxins must be tested. But if it is for oral administration, I don't need. So for the dosage form, if it is in the capsule or tablet form, usually we don't do the endotoxins. But if it is a sterile form, we do the endotoxins. Dissolution, I don't do dissolution for all dosage forms. I don't do dissolution test for liquid dosage forms like solution for injection or syrups. But I do dissolution for tablets, for example, and capsules. Friability is not done for capsules, but it is done for tablets. Okay, so this can be done for certain materials or certain dosage forms, not for all. The focus here is detailed performance and safety testing for particular applications, certain applications. It's for certain dosage forms or for certain methods of administrations or administration or certain substances. So it is a tailored to specific drugs or dosage forms like tablets, injectables, gels, and so on. I don't do viscosity for tablets, but I do viscosity test for gels and so on. These are examples. We can go through quickly. You don't need to, you just need to have a look and identify the most important ones. Those in blue color here are actually the most commonly used, like spectrophotometric identification. Usually you do IR to confirm the structure, infrared. Chromatography is used for assay mostly and for impurities. Mass spectrometry is used for, can be used for assay, can be used also for determination of molecular weight to confirm the structure. Spectrophotometry, light scattering is used for particle size determination. Spectrophotometry can be used also for like UV spectrophotometry for assay. Chromatography. So this for identification. Then we talk about probably spectrophotometry is about if you do some NMR, IR, and so these things. Okay. For assay, I usually use chromatography. and I use spectrophotometry and light scattering. Mostly chromatography is used like HPLC. Can also use UV. UV is usually used for raw material. Chromatography is used more for finished products. For impurities, still as I mentioned before, chromatography is routinely used, like HPLC. Spectrophotometry light scattering is not much. For residual solvents, I can also do residual solvents. I use chromatography, loss on drying. This in case of inorganic materials, I can use like elemental impurities limits test, as I mentioned before, like cadmium and so on. Toxic heavy metals can be determined. Mercury as well. Okay. Specific tests here, like for particle size, light diffraction measurement. Bulk density and tap density, this one used usually for determination of powder flow, and the other things you can go through. Can you mute, please? Okay. Last thing, we talk about excipient monographs before the last thing. Now, if I want to, can you mute, please, all of you? We have two important references that I can refer to if I want to get some information about my excipients. One is non-regulatory, just like a compilation of information about different excipients. It has monographs. We call it Handbook of Pharmaceutical Excipients. This is a very important reference. If you work in R&D later as a formulation scientist, then this is a good reference to know about excipients. This Handbook of Pharmaceutical Excipients contains monographs for different excipient types. Okay. And then in each monograph, you will find an information about information about the drug, the excipients. What information you can find? You can find different names, non-proprietary names, chemical names, commercial names. You can find empirical and chemical formulas, molecular weight, pharmaceutical specifications, chemical and physical properties, incompatibilities, and interaction with other excipients, which is very important. I want to start to make a formulation. I want to ensure that the excipients I will use in my formulation are not incompatible with my drug. So I can refer to this reference. I can see whether there are reported incompatibilities between this kind of excipient I want to use and between my drug and or no. If there is incompatibility, I should exclude. If no incompatibility, I can use. But still, I need to test to confirm as well. I can know about regulatory status. This is FDA approved, FDA not approved, whatever. Okay. There are information about safety, stability, and handling information, storage conditions, for example, and applications in pharmaceutical formulation or technology. What's the use of this one? It is used as adherent, anti-adherent, disintegrant, suspending agent, whatever, it will be mentioned in the book. And this is an example of the monographs. Like this one, I refer to high promillose monograph. I will find non-proprietary names. So it has different names. In British Pharmacopoeia, they call it high promillose. Japanese Pharmacopoeia, the same thing. Pharmacopoeia in USP and whatever, same name. Okay. And then there are any other synonyms. There are other synonyms. It's called also methocell, methyl cellulose, propylene methyl cellulose, propylene glycol ether, methyl hydroxypropyl methyl cellulose. So you have different names. You have the chemical structure. You have empirical formula and molecular weight. Functional category used as bioadhesive material, coating agent, controlled release agent, dispensing agent, dissolution enhancer, and so it's mentioned, all mentioned. What are the applications of this part? Okay, application, functional category, and applications of pharmaceutical technology. And they mention here that in applications they give more details. For example, what concentration is used for certain application, what viscosity grade you use, and so on. Okay, and you have also the so many other characteristics. They will give you the different grades available. We have different grades with different viscosities and so on. Nominal viscosity for the different grades. We have methocell, we have here K, we have E, we have F, we have other numbers as well. And each grade of them, it has different viscosity. So it will give you so many information about yours. But for compendial requirements and the regulatory side, then you have to refer to the compendia like USP or like European Pharmacopoeia and so on. So there will be a monograph for the substance in USP, for example. Here, it's not about the applications and so on. No, here it's about this substance, how to be tested, and what are the required characteristics to be approved for pharmaceutical manufacturing. It contains a name, description. It's, for example, white powder, granules, whatever, yellow color, whatever. They mention the physical characteristics. How to identify, what test to do to identify that this is the correct substance when you receive it from the supplier. How to assay, what's the assay method to determine the purity. What are the impurities that are usually in this substance and how to determine their amount, what's the method, and how should be the packaging and storage conditions. And any specific tests needed to better describe and control an excipient. For example, if any microbial limit required, sterility test, pH, viscosity, whatever, they will be also mentioned. This is an example of monograph in the pharmacopoeia for lactose monohydrate that we use as tablet diluent. So you have here definition, identification by infrared, thin layer chromatography, how to do, they give you the procedure, and then impurities, how to determine them, and then residual ignition, for example, heavy metals, and so on. And sometimes they don't give they give the method, some general tests, they refer you to the general chapters. Okay, and here specific tests to be done to this substance as well. And here, every test, they will give you the acceptance criteria. For example, here, acceptance criteria is a red color develops. If red color develop, then pass the test. If red color did not develop, then fail the identification test. It's not lactose. Okay? Everywhere they will give you the acceptance criteria. For example, heavy metals, not more than MT, 5 parts per million. If more than 5 parts per million, then your substance did not pass the test. If less, then it's accepted. Okay? This is another monograph you can go through and then you can see the tests. All these characteristics usually have impact on the final characteristics of the dosage form. For example, physical properties of particulate solids, mean solids that has in the form of particles. Particle size distribution, surface area, the flowability, density, wettability, hygroscopicity, and crystal state. They are all affected by these. They can be all tested by these tests, analytical sieving for particle size distribution, and laser diffraction can be used. Gas adsorption can be used to determine the surface area. Flowability can be by powder flow tests and so on. We test them because they have impact on the final characteristics of the dosage form, like weight and content uniformity, hardness and compressibility, disintegration, dissolution, and physical and chemical stability. For example, hygroscopic substance that will absorb water and then water content can increase degradation of the active ingredient. The wettability of the excipients affect the dissolution at the end. Better wettability than better dissolution. Less wettability, problem in dissolution. The things like density or porosity, and they will affect the hardness of the tablets, for example, and compressibility of the tablets, and so on. That's why you need to test them. The last issue in the lecture here is about can compendial monographs provide test for functionality? Means based on the tests I mentioned in the monograph, can I expect that, for example, lactose will have good flow or no? can I expect that lactose will have good compressibility or no? This cannot be known. So the current regulatory environment and the paradigm of quality by design go beyond simply identifying excipient function and emphasize performance through identification, evaluation, and control of critical material attributes. So I must do a study to expect how it will perform its function, how it will affect the characteristics of the dosage form. Then I should do quality by design. I have input the critical material attributes. I use, for example, substance with different grades of viscosity. I use, for example, different amounts of the substance in the formulation, whatever, and then I study the effect on the critical quality attributes of the dosage form. So I study the effect of critical material attribute on the critical quality attributes of the dosage form. Okay. Not all performance-related critical material attributes of an excipient may be identified or evaluated by specific tests and specifications listed in the pharmacopoeia. Cannot. I have to do my own study. I have to do my own study and then determine the characteristics of impact of characteristics of formulation on characteristics of the excipient on the characteristics of the formulation. You can refer to a chapter. I uploaded it to you on Caleb already, chapter 1059. This one, USP, in the USP, this chapter, it's about excipient performance and provides overview of typical material attributes for many functional categories, along with additional tests that may be useful to evaluate and control excipient attributes that are not typically included in the monographs. Other attributes not may be covered by monograph. Selection of the appropriate tests and specifications that are necessary to ensure consistent and reliable excipient performance requires understanding of the formulation and manufacturing processes, dosage form performance requirements, and the physical and chemical properties of each ingredient in the dosage form. So, for example, I'm preparing tablets. Tablets, for example, are immediate release. The performance or quality attributes are, for example, disintegration, dissolution, and friability. So these are the output variables. So I should know how much should be dissolved in 30 minutes, for example, how much should be the maximum for friability, how much should be the minimum, how the maximum time to disintegrate, and then I should do a quality, I should make a quality by design study, a design experiment, and then I should study the effect of characteristics of excipient on these characteristics of the tablets. Here an example, very simple example, magnesium stearate. We mentioned before that it is a functional, it functions as anti-adherent and lubricant for the tablets. We found that the surface area influenced the lubrication properties of magnesium stearate. Surface area is affected by particle size, as we know. And then this can impact the product properties. Here, the tablet tensile strength or tablet hardness. Tensile strength is hardness. The mixed study, they found that we have magnesium stearate from different suppliers, have different particle size and have different surface area. It doesn't, and then in the monograph, there is no specification for the particle size. So nothing about the functionality here of or lubricant effect of magnesium stearate. And they may also, this may not apply to other lubricants. Other lubricants may not have this effect of surface area or particle size on its function. Here in the study, they studied the tensile stress, means hardness of the tablets, relation with the lubricant surface area. So when the lubricant surface area increase means particle size reduced. What happens to the hardness? The hardness decrease. Okay, and this is actually, they don't decrease in the same pattern from all the suppliers. So for one supplier, at certain particle size, we have higher compressibility, higher hardness, and then decrease quickly. Some other suppliers, when we use magnesium stearate in the same amount in the tablet, we result in tablets with less hardness, right? So we have, it's different from one supplier to another. Last thing is excipient impurities and drug excipient interactions. As we mentioned, that excipients may interact with drug, and it's also impurities of excipients may interact with the drug and may cause maybe physical, maybe chemical interaction that results in inactivation of the drug. So they have some reactive functional groups. Here we talk about chemical interaction. Impurities originate from the manufacturing process. So maybe they are a side product, and may be also they are degradants from excipient agent. So impurities are two types, maybe side products during the synthesis and may also be degradation products. Impurities, if they have reactive species, reactive functional groups, like peroxides and aldehydes, they can interact with drugs directly and cause significant drug product stability issues even when they are present in low levels. And in this table, you don't need to memorize, just I want you to go through just to see. Here some drugs, and then here some impurities. And this is the excipient that contain the impurities. So for example, povidone and copovidone may have some peroxides, uh, and they cause some, they interact, okay? Uh, they interact with the drugs. If the drug loading is within this amount, then there is a possibility for interaction. There is a possibility for interaction, okay? Because usually impurities are in small amounts, right? So it depends on drug loading. Also, what is the amount of drug, the formulation, then the little the drug, then the more significant the interaction. That's the end of today's lecture. Just an introduction about excipients. It doesn't prepare you to be a formulation science directly, but at least we give some basic concepts that you have to understand that when you plan to prepare or to make a formulation, then what ingredients to. You'll study more about this in specific lectures about emulsions, creams, tablets, and other specific formulations. But at least you know that what references you should refer to when it comes to information about excipients, what incompatibilities they have, what different grades available in the market, what tests you need to perform on excipients, and so on, and what are the different functions excipients generally perform in the dosage forms. Any questions? Any questions? Okay, so we reached to the end of the lecture today. See you inshallah another the coming lecture or in the coming practice. Assalamu alaikum.